Hi, everyone. This is Miss Lasar, and I am here to tell you about your next topic, the cell cycle, starting with these amazing comics. Take a moment, pause the video, read the memes. It's worthwhile. Okay, so here's what we're going to be answering today, our lesson questions. Question 1, why do cells need to grow and divide? Question 2, how do cells grow and divide? And question 3, what happens if something goes wrong? This is a reminder during the lecture, you're going to be taking handwritten, super complete notes using the template that your teacher has given you. So let's start with the first question. Why do cells need to grow and divide? And you'll notice as we go through this slideshow that it's going to be a little bit more than just writing the text that's on the screen, because this slideshow is mostly pictures with just a little bit of text. And so you're going to need to listen to what I'm saying very carefully, rewind to parts when you miss something that I say, and write down not just what's written on the screen, but also what I'm saying, what I'm explaining about what's on the screen. Okay, so here's our first question. Why do cells need to grow and divide? Well, two reasons. The first reason is to allow you to grow. As you grow and get physically bigger, your cells don't just get bigger, we actually need more of them, because just like to make a bigger house, you need to add more bricks, not just expand the existing bricks. To make a bigger human, you need to add more cells. The second reason that cells need to grow and divide is to repair you. When you get a cut and you damage or kill some of your skin cells and you lose some of your red blood cells because they escape out of you as you bleed, your body needs to repair those broken cells and replace the cells that you lost. And to do that, your existing cells need to grow and need to divide into new cells. So question 2, how do cells grow and divide? Cells grow to about twice their original size and then divide in half. And let me show you what I mean. Here's a cell. This is a typical human cell. It looks nice and squishy. It's got a nucleus. It's going to grow to about twice its original size, and then it will divide in half. And notice it kind of pinches itself into and splits into two cells. And the purpose of today's lesson is to tell you a little bit more detail about this process, and then we'll take the different parts of this process, and in later weeks we'll learn about them in even more detail. Today will be an overview of this whole process. This process has to be very tightly controlled because the final cells need to be identical to the starting one. This original cell and these two final cells need to be exactly the same. What could go wrong? Why might they not be identical to each other at the end? Why is that a problem? Well, here's a picture of a dividing cell. Now you can see some of the parts in it. The DNA has been highlighted in blue. These other gray structures, these are organelles like endoplasmic reticulum, the mitochondria, ribosomes. We'll talk more about those later. You've learned about those organelles in the past. Check out those notes if you forgot what any of these structures are. And notice right here we have our cell pinching off in the middle, but it looks like the DNA is right in the middle of this pinch zone, and it looks like there's a pretty good chance that we lose some of this DNA because it gets like broken in half and torn apart as the cell is trying to divide. This would be really, really bad. This would cause DNA damage. Our cells at the end may end up with a partial set of instructions. They may end up with a set of instructions that has rips in it. That is a really big problem. Another thing that might happen is our cellular machinery might be divided unequally. So maybe we've got our DNA going in separate halves. That's good. One set of DNA to the top cell, one set of DNA to the bottom cell, but all the other organelles are like clumped down here in the bottom cell. And this bottom cell ends up with way too much cellular machinery. It's too big to function. The cell on the top ends up with too little cellular machinery. It's too little to function. That would be a problem too. So we want to make sure that during cell division, the DNA and the organelles are evenly divided. Let's talk about this whole process. This whole process of cells growing and dividing is called the cell cycle. And again, we start with one cell. We end up with two cells that are identical to each other and to that beginning cell. Again, we start with one cell, we end with two cells. All cells in question are 100% identical to each other. The initial cell is called the parent cell, and the final two cells are called the daughter cells. Here's a graphic representation, a little picture of the cell cycle. And at this point, you may actually consider drawing this cell cycle on your note sheet. We're going to label the different parts of the cell cycle. We'll go into detail about each part, and you might choose to take up half a page or a whole page drawing this diagram and adding the details next to each part of the cell cycle. So the rough parts of the cell cycle are this chunk, which looks like it's about 3/4 of the cell cycle. This is called interphase, and this is the growth, the replication. This is the daily life of the cell. And then we have this smaller part, which is actual cell division. This is called mitosis, or cell division. Cells spend way more time in interphase than in mitosis, and you can see that. Look, most of the cell cycle is interphase, the daily life of the cell. Only a little bit of the cell cycle is when the cell is actively dividing. And I'll show you a video at the end of this lecture where you can actually see that. All cells spend way more time in interphase than in mitosis, and the amount of time that they spend in interphase versus mitosis depends on how frequently they divide. The more frequently they divide, the more time they spend in mitosis. We can organize cells by how frequently they divide, by the amount of time they spend in mitosis, from least amount in mitosis to most amount of time spent in mitosis. Least amount of divisions, or dividing very rarely, to most amount of divisions, fastest divisions, most frequent divisions. For example, nerve cells never divide. Your nerve cells finish dividing before you are born, and then they might be making connections still once you're out of the womb, but they are not dividing anymore. And then for the rest of your life, they don't divide, which is why a brain injury or a spinal cord injury where those nerve cells are damaged is such a big problem, because those nerve cells will not be able to divide and replace themselves. Similarly, heart cells essentially never divide. They divide before you're born, they divide a little bit after you're born, and then they stop dividing pretty quickly, and they don't divide for the rest of your adult life. Again, why a heart attack, damage to those heart cells, is such an issue. They can't divide to replace and repair themselves. Liver cells do divide, and your liver cells actually replace themselves entirely about once a year, once every 3 years. Skin cells divide more frequently. Your skin cells divide and replace themselves more frequently. Your skin cells replace themselves about once a month. Intestinal cells divide even more frequently. They divide every couple of days. And bacteria cells divide even more frequently. They divide every couple of hours. Okay, so let's label the parts of the cell cycle here. We've got the big parts, interphase and mitosis, but interphase is actually broken down into 3 separate parts: G1 phase, S phase, and G2 phase. And you can go ahead and label those parts on your diagram if you drew a diagram, highly recommended. And we're also going to talk about each of those parts in a little bit more detail right now. G1 phase is the general growth phase. So now we're going to start with, notice on my diagram, here's where it is in the cell cycle, highlighted in green. We're going to start with a diagram of a cell, and you're welcome to draw this along with me. It's not super complicated. This cell is small. It has some organelles. It's got a nucleus with some DNA inside of it. Looks like an endoplasmic reticulum, some mitochondria, some ribosomes, other organelles that I didn't represent on there. And during G1 phase, the organelles are duplicated and the cell grows significantly. So by the end of G1 phase, our cell looks like this. It's much bigger. And instead of 1 endoplasmic reticulum, now I have 2. Instead of 2 mitochondria, I have 4. My ribosomes have been duplicated. But there's something that has not been duplicated: the nucleus and the DNA. I still just have 1 nucleus and 1 set of DNA. That is not duplicated in the G1 phase, but all the other organelles are. So at the end of G1 phase, we finish with our big cell with our organelles that are duplicated. And then we move into S phase, which is this green highlighted part of our cell cycle. In S phase, again, here's what we're starting with. A big cell, organelles are duplicated, but we still have one set of DNA. And now in S phase, our DNA is replicated. Replicated and duplicated mean the same thing. So our cell will look very similar at the end of S phase. The only change is that now I have 2 copies of DNA in the nucleus instead of 1 copy. And there's a note down here. This is really important. Note, DNA replication always introduces errors. Those errors are called mutations. And if you think back to our cancer lessons, you know that's a big problem because mutations in DNA is what changes the instructions of DNA and can lead to things like cancer. So we got to fix those errors. And in fact, G2 phase, the next phase of our cell cycle, here it is, this green highlighted part of our cell cycle. This is where we fix those mistakes that we made during cell division. All right, so we start G2 phase with our big cell. Organelles have been duplicated, DNA has been duplicated, and G2 phase is where we prepare for division. During G2 phase, our organelles for division, which are called centrioles, will be made, and our DNA errors will be fixed. Those mistakes we made during DNA replication, we will fix those in G2 phase. So at the end of G2 phase, our cell looks pretty similar. It's not a lot bigger. We do have these guys, centrioles, they have appeared, and our DNA is now fixed up. There shouldn't be any or many mistakes left in it. And at the end of G2 phase, we move into M phase, this last small part of our cell cycle. Here's the cell that we start with. This is what we ended with at the end of G2 phase. Our DNA has been duplicated already. Our organelles have been duplicated already. Our cell is nice and big. It's got these centrioles, and now we're ready to divide. And I'm going to show you how division works in a series of pictures. Feel free to copy down some parts of these pictures with me. The first thing that happens is the nucleus disappears. So our nuclear membrane literally disintegrates. The 2 sets of DNA are released, and they start separating themselves to opposite sides of the cell. So do the organelles. Then our cell starts pinching itself in the middle to separate these 2 sets of DNA and organelles. And eventually that pinch will be complete and we'll end up with 2 separate identical cells that look just like the cells we started off with in G1 phase, because these cells are now back to the beginning of our cell cycle in G1 phase. And so what I'd like you to do is represent those rough steps of mitosis. Rewind the video if you want to hear me re-say it. But just know that we're going to have a separate week on the details of mitosis. So if you're wondering, like, how do these things separate themselves? How do they get pulled to opposite sides of the cell? We'll answer that question in a later week. And we're in our final section, section 3. What happens if something goes wrong? Well, so if something goes wrong, that's a really big deal. We could end up with DNA damage. We could end up with cells that are not functional. So we have checkpoints in place to make sure that nothing goes wrong. Just like a stop sign or a crossing guard, just like a security checkpoint at an airport, they are there to make sure that everything has gone to plan, that everybody has the credentials they need, is going in the right direction, and is ready to move forward in the process. Let's talk about the cell cycle checkpoints. So there's going to be 3 checkpoints. I'm going to illustrate them on my diagram of the cell cycle and tell you the details of each. Our first checkpoint happens at the end of G1 phase, right here where this red line is. This is called the G1 checkpoint. So think about what's happened at the end of G1 phase. Our cell is much bigger. It should have duplicated its organelles. And the purpose of the G1 checkpoint is to check to make sure that that's happened. The checkpoint is making sure, is the cell large enough? Are there duplicated organelles? And is the cell still receiving growth signals? If the answer to these 3 questions is yes, then the cell passed and it will move on to S phase. If the answer to any of those questions is no, the cell fails the checkpoint and it goes into G0 phase, which is a new phase that I haven't told you about yet. What the heck is G0 phase? G0 is a pause in the cell cycle. In G0 phase, the cell exits the cell cycle and it just chills out there. It continues its normal life functions, but it stops all moves toward dividing. Most cells spend some time in G0 phase. This is where they are when they're not actively growing or preparing for division. Nerve cells and heart cells, cells that never divide, spend pretty much all of their life in G0 phase. And that's why they don't divide, because they're not able to leave G0 phase and move on to the rest of the cell cycle. But other cell types will be able to leave G0 phase at some point, return to the end of G1, pass the G1 checkpoint, and move into S phase. So let's say our cell has done that. It's moved through S phase. It replicates its DNA. There's no checkpoint at the end of S phase. It moves through G2 phase. We fix those errors. We make the centrioles for division. At the end of G2 phase, we hit our next checkpoint. This is the G2 checkpoint. The purpose of the G2 checkpoint is to make sure that things have gone correctly. Are there any mistakes left in the DNA? We need to make sure that there are no mistakes left. If the cell can verify that there are no mistakes left in the DNA, the cell has passed the G2 checkpoint and it will go on to M phase. But if there are mistakes left in the DNA, the cell fails the G2 checkpoint and it will self-destruct. It won't just pause. It won't just wait. It will self-destruct. Let me tell you a little bit about that. If we haven't passed the G2 checkpoint, that means there's major DNA damage and the cell does not have the correct instructions to be a functional cell. The cell will go into programmed cell death and it will self-destruct. Programmed cell death is also known as apoptosis. Here on the left is a cell with too much DNA damage. It looks just the same as a normal cell, but inside the nucleus, the DNA has been ripped up. There are errors. We can see those errors, and it hasn't been able to fix them. The cell will self-destruct, and it looks kind of like this. The cell will actually fall apart and will dissolve into smaller pieces. Then neighboring cells will absorb those smaller pieces and use them as building blocks to add on to themselves, grow, divide, and create new cells. But let's assume that we passed the G2 checkpoint and we move on in the cell cycle. We get into mitosis. And at some point in the middle of mitosis, we will hit the M checkpoint. The purpose of the M checkpoint is to make sure that DNA is going to be split evenly between the daughter cells. So remember this picture that showed that the DNA and the organelles were going to be like dividing themselves equally between the top half and the bottom half. That's really important because each daughter cell needs exactly the same DNA and exactly the same amount of DNA. So we need one set of DNA to go to the top cell, one set of DNA to go to the bottom cell. And the M checkpoint is making sure that that happens. If the cell passes the M checkpoint, if it can verify that the DNA will be dividing evenly, it will complete M phase and move on back into G1 phase and continue through the cell cycle. If the cell fails the M checkpoint and cannot verify that the DNA will be divided evenly, it will go into apoptosis. Remember, apoptosis is that programmed cell death. And that's because it means that the daughter cells will not be getting the right amount of DNA. One might be getting more than the other and will not have the correct instructions to be a functional cell. So the cell will go into programmed cell death and it will self-destruct. Okay, the last thing I'd like to show you is real cell division. Here it is. These are cells that are growing and dividing in a petri dish under a microscope. We start with one cell and it's dividing. It's in mitosis right now. You can see it scrunching up and now it is 2 cells. Okay, so they just divided. That means they're back in G1. You'll be able to see them grow through G1 phase, and then we'll see them go through S phase, go through G2, and then eventually go through M phase and divide again. These are actually a type of human cancer cell that are frequently used in biology experiments because they grow much faster than normal human cells and they divide much more frequently. They divide almost as frequently as bacteria cells. They're growing in a petri dish under a microscope right now. And so we're filming them. And this is definitely a sped up video. This is happening over many hours, not the seconds that you see. But it is a type of cell that grows and divides very frequently. You can see these cells are much bigger than they were when they just divided. And pretty soon, speed up the video. There we go. Pretty soon you'll see them. They're much, much bigger. Oh, this one on the right is dividing. Wow, that was super fast. It is definitely sped up, but notice how fast that division was in relation to how it grew. It took much, much, much longer time, much more time growing than it did dividing. And now we've got 4 cells that are back in G1 phase. And so that's what real cell division looks like. And I hope you learned a little bit. Make sure those notes of yours are super complete. Take a picture, upload them. Thanks for watching this video, and we'll see you next week.